regulation of the cell cycle is very much essential for a particular cell that undergoes the division, because when a cell undergoes the division, it produces various several kinds of proteins and enzymes that will help the cell to undergo the division or complete the division. If there is no regulation and there is a problem during the synthesis of those particular proteins and enzymes, the cell ultimately converts into a cancerous cell. And if there is a regulation, the cell will go to apoptosis. That means the programmed cell death, that is the killing of that particular cell. Hello friends, welcome to Biology Concern. I am Animesh, and in this video I am going to discuss about the regulation of the cell cycle in molecular way, that you can understand the detailed mechanism of each of the particular protein, enzymes and the phases, particular certain phases of the cell cycle very clearly in a short time. Okay, so let's jump into the topic. First up thing, the discovery of the cell cycle regulation. In the year 1970s, a series of cell fusion experiments done by Potu Rao and Robert Johnson at University of Colorado and helped to open the understanding of the cell cycle is regulation. Okay, so their work, Potu Rao and Robert Johnson, what they did, they actually did fuse the cell G1 cell to M cell. That means a cell that is the condition in the G1 gap 1 phase, and another cell that is in the M phase, that is the mitosis phase. They fuse two cells together. Next they fuse S cell, that is the synthesis phase cell, to M phase cell, and G2 phase cell and with the M phase cell, and they conduct a series of experiments that will lead us understanding how the cell is regulated. And Leonard Hartwell and Ted Wynyard in 1988 concluded that cell processes checkpoint as the part of their cell cycle. So if we considered as a cell, and if this is a cell cycle, 10% of this is the M phase, okay, 90% of this interface, this one is the G1 and S and G2. So there is 3 checkpoints are there. One in G1S checkpoint, another one is G2M checkpoint, and third one is the M checkpoint or spindle assembly checkpoint. So we observed here 3 kinds of checkpoints are there, and those restriction points helps to determine the cell fate. If any protein or enzymes is malformed or malfunctioning during this time of cell division, then the cell ultimately leads to the apoptosis, that is the programmed cell death. Okay, so leads to the apoptosis. If all the things, all the proteins and all the molecules are function properly, then the cell cycle continues. So this is the basic hypothesis, and this is the basic concept about the cell cycle regulation. Here in cell cycle regulation, several proteins and several enzymes are performed together to conduct those cell cycle checkpoints and cell cycle regulation in a particular manner or precise manner. One of them is MPF, that is stands for maturation promoting factor. Maturation promoting factor. Maturation promoting factor helps the cell to lead from G2 to M transition. So the entry of cell into M phase is regulated by a protein called MPF, that is the maturation promoting factor. So maturation promoting factor ultimately helps the cell from G2 to M transition, and here very much essential, MPF is consisting of one of the protein and the enzyme. The protein is cyclin and the enzyme is CDK, that is the cyclin dependent kinases, and those cyclins and CDKs present throughout the cell cycle, throughout the proper cell cycle. Okay, and here I gave a chart of the cyclins and CDKs that will help the cell to undergo or to complete the cell cycle. In early G1, there is a cyclin D activated along with CDK4 or CDK6. In late G1, cyclin E is activated with CDK2. In S phase, cyclin A is activated with CDK2, and in G2 phase, cyclin B is activated with CDK1. So these cyclins and CDKs perform a crucial way or perform a crucial task of the cell cycle or regulation of the cell cycle. How? First of thing, we know about the kinase or CDK, cyclin dependent kinase. So CDK is a kinase. CDK is a kinase that transfer a phosphate from the transfer of phosphate group from ATP to specific serine or threonine residue of a particular protein. So in case here the CDK actually transfers a phosphate group from ATP to a particular serine or threonine amino acid or threonine amino residue of a particular protein, and the regulatory subunit that is called cyclins. So in case here when a cell in early G1 stage there is cyclin deactivated along with CDK4 or CDK6, and in late G1 cyclin E is activated along with CDK2. So when the cyclin D is activated, here I draw a diagram that is conduct briefly that regulation of cyclins and CDKs, that is the upregulation and down regulation of cyclins and CDKs that performs in a cell, inside of a cell or inside of each particular checkpoint. So in each particular checkpoint this total regulation is occurred. Okay, and we are gonna take a look about this particular diagram in a short moment of time. But before, we have to know that those cyclin CDK complexes are very much important for progression of a cell, for progression of a cell from one phase to another, and a transition from G2 to M. That is very much crucial point is here. Okay, so first of thing that cyclin is a regulatory unit. Okay, that is the protein, and CDK is a cyclin dependent kinase. It is actually a enzyme that will help to bind with cyclin and activate their product. Okay, so first of thing, if we consider about the G1S transition point, that is this checkpoint, so we found that at first CDK is present there. When there is no cyclins, CDK is not activating, CDK is inactive. In this time CDK is inactive. But when cyclin is present, or building up the cyclins inside of the cell, then cyclin binds with the CDK along with inhibitory kinase, that is the V1 activates, and V1 actually phosphorylates. V1 is actually a inhibitory kinase. So V1 is a inhibitory kinase. So first of thing, it is a kinase, that means it transfer phosphate from one ATP to specific serine or threonine or tyrosine residue. And next thing is that it is particularly inhibitory in nature. Inhibitory in nature means, that means when V1 is present, the whole complex is still inactivate. Okay, so in case here CDK, the tyrosine of 15 number residue is phosphorylated. Here you clearly observe that the 15 number residue of tyrosine is now phosphorylated, and we have got a cyclin CDK complex along with the 15 number residue of tyrosine is now phosphorylated. Second thing is that CAC is there. So next thing, next important kinase is CAC. The full form of CAC is CDK activating kinase. So this CAC is activate this particular CDK, and here its CAC activates or phosphorylates the threonine 161 residue. Okay, so it phosphorylates threonine 161 residue. In case of metazoans, in case of Saccharomyces cerevisiae, the CAC is activating 160 number residue. So there is a difference between Saccharomyces cerevisiae and human. In case of human, threonine 161 is activated or phosphorylated, but in case of Saccharomyces cerevisiae, threonine 160 residue is phosphorylated. Okay, so the complex is still inactive, right? Because CAC, because V1 is acting over CAC, that means V1 is more aggressive and V1 is inhibit the function of CAC. Next thing, another enzyme came into play, that is a CDC25, that is one kind of phosphatase. Those phosphatase ultimately removes the phosphate group from tyrosine. So here, from here, removal of the phosphate groups leads to the activation of these particular cyclin CDK complexes. Okay, so now CDK and cyclins are activated. In this case, when a particular phosphatase molecule, that is the CDC25 enzymes, are activated, now these cyclins and CDK complex are activated. And after the performing the total function, that is the completing the transition from G1 to S, now those complex now been dissolved or degraded. How it is done? It is done by actually a protein known as DBRP. DBRP full form is destruction box recognition protein. So DBRP full form is destruction box recognition protein. Okay, so what is meant by those destruction box? Each of the protein in its N-terminal end, there is a specific 9 residue complex known as destruction box sequence. Okay, so in the N-terminal of particular each protein, in case here is cyclin, the 9 specific sequence is known as destruction box sequence. So those DBRP, destruction box recognition sequences, those proteins are actually search whole the polypeptide chain, and it ultimately finds the N-terminal residue of specific 9 sequences, and then it binds with that particular 9 residue sequences and activate SCF, that is the escape equivalent 1 F box containing complex, that is a E3 ubiquitin ligase. I told about SCF in my previous video. So SCF, that is the CDC25, again works, and those SCFs are actually ubiquitinize the whole cyclin, because whenever a molecule or a protein needs to be destructed, needs to be degraded, the protein should be passed over a large complex known as proteasome complex. Okay, so known as proteasome. So if a protein is passed through a proteasome complex, a proteasome is only recognizes the protein which are actually ubiquitinized. Ubiquitin is a small molecule that is present in each and every cell, inside of each and every cell, and those ubiquitin molecule are bind with those particular 9 sequences, that is an N-terminal of particular each and every protein, and those sequences known as destruction box sequence, and ubiquitin attached with N-terminal of those particular destruction box sequences. And now those cyclins are passed through the proteasome complex, that is a large complex, proteasome complex, and now cyclin is now being degraded, and when the cyclin is now being degraded, CDK is still free, and now CDK is perform again another cycle of particular thing. Okay, so this is the total mechanism of each checkpoint. So here at first cyclin D is activated along with CDK 4 or 6. Now it is time for destruction or degradation of cyclin D. So now cyclin D passed through those particular proteasome complexes with the help of SCF molecule, with the help of SCF. So from G1, this is the M, CDK, cyclin D and CDK 4, 6 now been degraded. Okay, so at first cyclin D is ubiquitinized, and in late G1 it passed from a proteasome complex and now been degraded. Now cyclin E is synthesizing, and CDK2 is also present inside of the cell, and cyclin E is combining with CDK2, and it promotes the cell from past the restriction point, that is the G1S restriction point. Cell is now passing from the G1S restriction point. After that S CDKs or S CDKs is present here. S CDKs also has CDK2, and S cyclin is present. S cyclin is now building up inside of the cell, that is the cyclin A. So cyclin A and CDK2 are now made a complex, and it helps to progress from S to G2 phase. Okay, here cyclin A and CDK2 complex now being degraded with the help of those mechanism I already discussed about the mechanism here, and now cyclin B and CDK1 is activated. This cyclin B and CDK1 leads the cell to pass from G2 to M transition, and it holds the cell until the next G1 of the cell. So here is a simple chart of the complex thing here. That is a G1 is in blue color, that is a G1S cyclins, red color is the S cyclins, and green color is the M cyclins. Okay, so here in case of G1, that is the start point. Here G1S cyclins are higher. The concentration of G1 cyclin is higher. G1 cyclins means G1 cyclin mean cyclin E. Okay, G1S cyclin mean cyclin E because it is consists of late cyclin, late G1 cyclin. That is the G1S cyclin is the E cyclin. So E cyclin is present here now build up in the inside of the cell and it combines with the CDK2 and it proceeds to the cell from G1 to S phase. Now S cyclins are the concentration of S cyclins in G1 is low, very much low, but when it progresses towards the S phase, those S cyclins or cyclin A is now built up inside of the cell and now its concentration building up and it combined with cyclin A with CDK2 and it helps to progress the cell from S to G2. In case of G2 phase, here G2 to M transition, here the concentration of M cyclins, that is the cyclin B, here cyclin B rises its concentration and it binds with the CDK1. So cyclin B and CDK1 binds up and its concentration is higher and it helps from to pass a cell from G2 to F transition. Okay, after that it again repeated its in this manner and completed those processes. So this chart is depicted here. Each of the restriction point works, how each of the restriction point works. That means when CDK is alone, CDK is inactive. When cyclin is present, CDK is complex with the cyclin CDK complex. But here inhibitory kinase V1 acts as and phosphorylates 15 number residue of tyrosine and it's still inactivate. After that CAC, that is the CDK activating kinase, that is building up inside of the cell and that helps the phosphorylate threonine 161 residue and it ultimately inactivate because tyrosine 15 is still phosphorylated, okay, and V1 is still present. After the removal of V1 from the tyrosine and those tyrosine are now being dephosphorylated with the help of series 25. This is a phosphatase molecule, this is a phosphatase enzyme. Now the total complex is activated. After performing the transition phase, this total complex, cyclin CDK complex, now being degraded with the help of DBRP protein. That is, DBRP triggers the addition of ubiquitin. So SCF here comes into play and it ubiquitizes the cyclin and those cyclins are now passed through the proteasome complex and those proteasomes are helped to degrade those cyclin molecules and CDK is still inactivate because if there is no cyclins, CDK is still inactivate. If there is building up cyclins more and more, concentration is more and more cyclin, so then CDK is activated. So each of the CDKs, all of the CDKs, CDK4, 6, 2, 2, 1, all of the CDKs present inside of cell, but they are not activating because there is no cyclins. If there is a certain cyclin present, then certain CDKs are activated and those CDKs helps to pass through the transition from G1 to S, S to G2 and G2 to M. So this is the complete regulation of the cell cycle. I think this will help you to understand about the complete regulation of the cell cycle and the molecular events occurred during the each cell cycle. If you like that video, please hit the like button. Don't forget to subscribe. Thanks for watching.